Welcome to BioInfoBytes. Let's get these formats decoded.
So, you know that feeling, right? You've spent hours building what you think is the perfect bioinformatics pipeline. You've got all the right tools, your data's ready to go, and then it just breaks. Well, 9 times out of 10, the problem isn't your code. It's actually a language barrier.
You see, in the world of bioinformatics, every tool speaks its own language, and that language is the file format. If you give a tool the wrong format, your whole pipeline can grind to a halt before it even starts. So today, we're going to decode the 3 most essential biological file formats you absolutely need to know.
All right, first up, let's talk about the absolute workhorse of bioinformatics, the FASTA format.
Think of FASTA as the universal language for sequence data. It's incredibly simple and clean, which is exactly why pretty much every bioinformatics tool on the planet can read it. Its only job is to store sequence data. No extra baggage, no complicated metadata, just the sequence.
And when I say simple, I mean it. Take a look. A FASTA entry literally has just 2 parts. First, you get one single header line that gives you some info about the sequence. And second, right below it, you have the sequence data itself, whether that's DNA, RNA, or protein. That's it.
Now, here is the one rule you cannot forget. The header line has to start with a greater than symbol. No spaces before it, nothing. Everything after that symbol on the same line is your sequence ID and a description. The actual sequence then starts on the very next line. And what if you have a bunch of sequences? Easy. You just start a new entry with another greater than symbol. We call that a multi-FASTA file. Simple as that.
Okay, so if FASTA is like a quick text message with just the core info, then GenBank, well, GenBank is the entire annotated novel.
The GenBank format, which you'll typically download from NCBI, is so much richer. It doesn't just give you the sequence, it gives you the whole story behind it. We're talking about detailed annotations, where the organism came from, links to papers, and a map of important features, like genes and coding regions.
Just look at the difference here. Up top, the LOCUS field gives you the basics. You've got a definition and the accession, which is its unique permanent ID. But this section right here, the features section, oh, that's the gold mine. It annotates every important part. And then way down at the bottom, in the origin block, you'll finally find the raw sequence data.
Let's just quickly break that down. LOCUS is your high-level summary. The accession number is what you'll use to look up this exact entry forever. But seriously, pay attention to that features table. This is what makes GenBank so incredibly powerful. It maps out everything from coding sequences or CDS to regulatory regions, telling you exactly where they are on the sequence that's listed in the origin section.
So, we've covered sequences as these long, linear strings of letters. But biology isn't flat, right? What about shape and 3D structure? For that, we need a totally different kind of blueprint. Let's look at the PDB format.
PDB files are not about the linear order of things at all. They're about the physical, 3-dimensional architecture of a molecule. It's designed to hold the data from experiments like X-ray crystallography, giving you the precise X, Y, and Z coordinates for every single atom in that structure.
And if you peek inside a PDB file, you'll see it's basically a big table of numbers. After some general header info, you get to the most important lines, the atom records. Each line is one atom, telling you its name, what amino acid it's part of, which chain it's in, and, most importantly, its exact X, Y, and Z coordinates in space.
Inside the file, you'll mostly see those atom records for the standard protein bits, but you'll also see HETATM records. That's short for heteroatom, and it's for anything that's not part of the main protein chain, like a drug molecule or a metal ion. And of course, you're not meant to read this file yourself. You open it up in a molecular viewer like PyMOL or Chimera, and that's when the magic happens. It turns all those coordinates into a beautiful, interactive 3D model.
Okay, that was a whirlwind tour. We've covered a lot of ground. So how do you actually know which format to use for your project? Let's put them head to head.
Honestly, it all boils down to what question you're trying to answer. If you just need the raw sequence to pop into a tool like BLAST, FASTA is your best friend. It's simple and universal. If you need to understand the function of every gene and feature in a sequence, you've got to use a GenBank file for all that rich annotation. And for anything to do with 3D shapes, protein folding, or drug design, well, PDB is the only way to go. So let's nail this down one last time to make it super simple. Use FASTA for sequences, use GenBank for annotation, and use PDB for structure. Getting this right is going to save you countless hours of frustration and keep your pipelines running smoothly.
And that is your bite-sized breakdown on biological file formats. If this helped clear things up, make sure you subscribe. Join us next time as we dive into one of the most confused topics in all of bioinformatics, the difference between similarity, identity, and homology. You really don't want to miss that one. See you on BioinfoBytes.